You know, I don't really look like this. I just started the cameras and I make this goofy face when I make sure that they're both recording and I don't cut it out so that you can laugh at me. Hi everybody, it is I, your all-time favorite animated creature from beyond, Richard Blyle. Today we're going to talk about epigenetics. Epigenetics. This brought to you by the guy who does this podcast. This is called Redeeming the Past. It's a podcast. It's all about addiction. And you may not know this, or you may know this. Recent studies have suggested the possibility of a connection between genetics, which we talked about last time, and addiction. That being the case, that's actually why we started by talking about genetics last time. Today we're going to delve into epigenetics. He is going to have two experts on epigenetics on his show sometime in the near future. And he asked me to explain what epigenetics is, and I thought, you know what, if I'm going to explain it, I can explain it to all of you. Now, I am not an expert. I'm a chemist. I'm kind of a generalist. That's why I can speak on such a wide range of topics. I do a little bit of research, I understand what they're doing, things sort of click, and I can give you the background. But believe me, I am nowhere near the expert that his guests are. I'm just going to give you some background here on epigenetics. Now, we're going to review a little bit of genetics in doing this, because we kind of have to. But in order to understand this, we have to understand DNA. I don't even know how to spell DNA. It's a joke. I think a lot of you probably know that DNA is a helical molecule. And so people tend to assume that it kind of looks like this. But it's a double helix. So they tend to think that, well, then there must be another helix here somewhere going maybe this way. Well, that's not quite right. The reality is the two chains of DNA kind of chase each other. If you've ever seen two dogs running and playing, the one is always sort of right by the other, and that's kind of what the DNA kind of looks like. You have one chain. Kind of loops around. And then you have a second chain that is very close to it. Did I do this right? Well, we'll say that I did. I don't think this is quite right. If you really want to know, there are lots of good images of these. You can Google it, and it's fine. But what's important about this? A couple of things you need to understand. This particular structure gives rise to two grooves. We call them the minor groove. The minor groove is the distance between the two here, where they're very close to each other, versus the major groove, which is the large space in between. Now, last time we talked about the four nucleic acids, adenine, thymine, cysteine, and guanine. Don't confuse guanine with guano, because that would be batshit. And they always pair up. Adenine always pairs with thymine. cysteine always pairs with guanine. They're held together by what's called hydrogen bonding. That part is not so important for you to understand, but the way that they work is they fit nicely together. There are 3 hydrogen bonds that can be formed or accepted in cysteine and guanine. There are only 2 that can be accepted between adenosine and thymine. And they fit. So if you have adenosine here and thymine here, they fit like your fingers would fit. Just like that. The longer index finger matching up with the pinky, and it's just a perfect fit. If you try to do an adenosine, trying to do this so it doesn't look too offensive, it's not quite going to match up. It just doesn't work. You see? The spacing is wrong. It just doesn't work. My first instinct to show you this was to make the 3 hydrogen bonds look like this, but some of you might be offended by that, so we're not doing that. That's a different story altogether. Okay, so anyway, the base pairs are actually flat. The molecules themselves, there's rings of sugar, and they're basically flat molecules. And when you pair them up, they remain flat and lined up. So the nucleic acids stack like plates in your cupboard. But there's a twist to them. So they stack with a slight twist all the way up. That twist is because of the twist in the DNA, which I've not given myself enough space here to do. So you have plates. And if memory serves, one complete twist, if I'm recalling correctly, are 6 plates. 6 of these plates. Now, what is epigenetics? In epigenetics, they are trying to control the transcription of these nucleic acids from DNA into RNA. They're trying to stop that transcription. And there's a couple different ways they do this, but the interesting thing is, for the most part, now we're going to talk about one way to stop it that's not natural, but generally they're trying to mimic what Mother Nature does to stop the transcription. Because like I said last time, you don't need every protein mapped out in your DNA to be produced simultaneously. You don't need them all at the same time. By the way, this is way misleading because there's no blank space there. The molecule is much more compact. Seriously, look up an image of it, or look up a realistic image of it. Not just sort of the cartoon that people tend to draw. This is a cartoon as well. But look it up. Okay. So, in order to transcribe this, there are proteins. Kind of wrap around the DNA. And they spin with the same twist that the DNA has. And as these are going along, they are reading those nucleic acids. They're reading the codons. As you recall, a codon is a series of 3 of these amino acids. And each codon has a different meaning most of them represent one of the 20 amino acids in proteins. But there are 6 codons, which I actually looked up since the last time I made one of these videos, because I was sitting here hanging out looking like an idiot. Anyway, there are 6 codons. 3 of them mean start, 3 of them mean stop. So when this molecule is spinning along the DNA, reading these codons, if it reads start down here, it starts transcribing from the DNA to create the RNA, which, as you recall, the rRNA, the ribonuclease RNA, that's the factory. That's where proteins are built, in the rRNA. So when it reads start, then it starts transcribing these into rRNA. And it keeps transcribing until it hits a codon that says, stop, you're done. This protein, that's all there is to it. But how do you stop this protein from transcribing those codons? How do you stop it? Mother Nature has a couple different ways that it can do it. I'm going to explain these to you briefly. I'm going to give you a third way. It actually goes back to a research project that I was doing when I was a graduate student. We'll talk about that in a moment. But first of all, understand this. This DNA, in order to make you, this DNA, if you were to take all the DNAs and link them together to make one long chain, they don't work like that. They're split up into genes. So your DNA is actually split up into, is it 5, 6 genes, something like that, so pieces of DNA. But if you had one DNA that had all of your genetic code, that DNA would be over 5 feet tall. If you held it up, you would still not be able to see it because it's only one molecule thick and you can't see anything that small. It'd be like an invisible thread. So, here's the question. How is it possible for every cell in your body, and the technical term for the size of cells, I don't know if you're aware of this or not, the scientific, when we talk about cells, the technical term for their size is teeny weeny. That's a technical term, so don't let it confuse you. Itsy bitsy, teeny weeny. Alright? Your cells are wearing a yellow polka dot bikini. No, they're not. It's reference to an old song. You're too young to understand that reference. Okay, some of you may not be. I'm as old as the hills. I stood in line to get an autograph from the author for the very first edition of the Bible. That's how old I am. Okay, so anyway. What was I talking about? How is it that you can get all of this in one cell? Well, would it surprise you to discover that right now in my pocket I have 500 yards of thread? How is it possible for me to get 500 yards of thread in something as small as my pocket? If you so, you know the answer. It is wrapped around a spool. A spool. Of course, a spoon would make no sense. It must be a spool. So, in this analogy, this thread represents the DNA. And that's how your DNA is stored in your cells. The spool itself is a protein called histone. actually worked on kind of an interesting project that suggested that the way DNA wraps around a histone is by neutralizing the phosphate backbone, which is a very charged backbone. But anyway, that's neither here nor there. That has nothing to do with today's topic. So you have a histone, which is a protein, which is kind of like the spool. Then you have your DNA wrapped around it. So one way that your body, not just your body, your biochemistry, your metabolism, your catabolism, one way that your body can control whether or not proteins are transcribed is by what is on the inside and how tightly the DNA is wrapped around that histone. If you look at this spool, it is not hard to picture the fact that what you're seeing here is just the outside thread. It's all you're seeing is what's on the outside, which is clearly, although it may be hard to see in this light, it's brown. I wear brown pants because I scare easily. But is it brown throughout? You would hope so. You would assume so, but let's face it. This thread is wrapped around here many, many times, and there are different layers. So there is a chance that on the inside, maybe they ran out of dye. So maybe it's not brown throughout. That's kind of what your histone can do. It can actually turn the DNA so the start and the stop is on the inside. This protein is not going to see that. That's one thing. One way epigenetics, an epigeneticist could use that trick to their advantage if it turns out that those addiction genes really are real. What fascinates me about that concept, though, is if there are addiction genes, that means that there are addiction, not necessarily proteins, but something like proteins. Could be peptides, could be enzymes. Enzymes are basically, and what we're talking about here, by the way, in case you're curious, this is a little beyond the scope of this talk. Peptides are very short. Enzymes are longer, 20 to 50 amino acids long, but they're all made of amino acids. Proteins then are between 70 and 100,000 amino acids long. It's length, but the basic concept, the basic building structure is the same, so it's all right here in your DNA. Oops, I made a mistake. I accidentally erased part of the phosphate. Which brings us to the second method in which we can stop this gene from being transcribed. Your body will sometimes attach a little atomic species on the phosphate backbone. What it attaches, actually it's very easy to attach, it's very easy to remove. It has to be, because we want it to be a temporary block, not permanent. So it's temporary. Now this is a real chemical bond, it's a real chemical reaction. Yes, it releases energy when you attach it, it requires energy to remove it, but it can be done fairly easily with not a whole lot of energy. Typically, what your body will attach is called a methyl group, which is a carbon with 3 hydrogens. And it just bonds right to the phosphate backbone right there. And what happens? Your molecule is happily going along reading your codons, and all of a sudden it hits this bump in the road. Thump! Unexpected. If you've ever put a coin on a railroad track, shame on you. But what were you trying to do? You were trying to derail the train. Well, imagine welding a cannonball onto that railroad track instead. And all of a sudden, the front wheel of that train hits that cannonball, and guess what? You have a big mess. It's not going any further. Same thing happens here. So if your start codon is right here, and this is your addiction peptide, enzyme, protein, whatever it might be, as soon as this protein hits that block, it can't go any further. If it cannot go any further, that's it. It's done. These are two of the methods of which I'm aware. I think there might be a third. But these are two of the methods that I know of that your body controls what gets transcribed, what does not. There are artificial methods as well. This is a project I've been working on since around about 1990. It's like a 35-year project called distomycin. Distomycin is an anti-tumor drug. Now, if you think about what cancer is, what tumors are, you have a mass of cells that, just like any other cells, they divide. But the problem is, in tumors and in cancer, they are dividing out of control. They're dividing too quickly. Every new cell must have, whether it's cancerous or not, or healthy, every new cell must have a complete DNA record. So this little protein is sitting here reading and transcribing all of these nucleic acids so it can build a whole new DNA. Distomycin is what's called a sequence-specific binder. A sequence-specific, oh boy did I misspell that, specific binder. It binds specifically to the sequence AAATT. This should have been published. It's embarrassing that it was never published, because I figured it out. I figured out how it does it. My question was, how does distomycin recognize? Because it's a molecule. It doesn't have a brain. It doesn't have eyes. It can't see the sequence. So how does it recognize the sequence? AAATT. And I figured it out. But once it finds that sequence, AAATT, it binds in the minor groove. Let's say it binds here. Now, your protein here is not reading the minor groove, it's reading the major groove. But again, just like this methyl groups, it's sliding along, it has a little something in that groove holding it to the DNA. And if it hits something unexpected, if all of a sudden there's a block there, this time inside the groove instead of on the phosphate bond, so it's different. If it hits something unexpected, it stops. That's it. So there are molecules, organic molecules, that can be designed to bind to specific sequences that will also control. Now this is an artificial means of controlling transcription. The advantage to this is you can actually design different drugs, and my hypothesis proved that this you can design them for basically any sequence you want. ACCGT. Or any length you want. ACCGTTAC. You can bind all of those. So if you know the sequence you're trying to stop, just bind something to it and it can't be transcribed. That, my dear viewers, is what epigenetics is all about. This is not the details. I am not an epigeneticist. I'm not an expert. But now you have an idea. And my good friends over at, hopefully, Redeeming the Time podcast, hopefully they have a little bit of a background information so that when they have their epigeneticists on their show, they can ask intelligent questions. But as for me, I am out of here.